Good morning, everyone. Welcome to the session, Translational Biology. Reminding you, this is the last session of the HDR workshop for knowledge-guided machine learning, which is part of NSF HDR program. So this is the second year of the funding. And so we are in the last session on the translational biology. So hope to remind everybody to submit your question through the Slido channel. Okay, so today in this session, we have 5 distinguished speakers. And before we get to their speech, I'd like to make a short introduction in the session. So our focus is on knowledge-guided machine learning, in particular, how the knowledge can be transferred in the translational biology domain. And so in this, I hope to mention the one of the main issues in applying AI machine learning approach in the biology is the big P, small n problem. So as you all know, so the deep learning has been popularly used everywhere, including the biology domain. And it has been especially the very famous multi-omics data, how to integrate multi-omics data for different disease predictions. But we are facing the huge problem is the small samples, but very large dimension and features. And some of the features may not be complete. So that I bring you the famous TCGA data sets. You can see they have 33 different cancer types with 20,000 primary cancer samples. However, if you look into different types, each type of cancer only has a few hundred samples. And the best we have is a little bit over 1,000. So definitely this is each type is not sufficient, the number of samples to be used to train a deep learning model. So that's the problem we are facing, how to apply machine learning effectively in small samples. So with that, I hope to introduce the latest machine learning community. So with the machine learning community also realized this problem for big data and small, long tail for small data problem. So a lot of research have been, working on this few shot learning problems. So with that, we have, so the biggest problem we have here is how do we see just a few patients and with different diseases, and then giving you a new patient, we can detect whether a new patient has what disease. So it's called a few shot learning. And so with that, what we general idea is to transfer knowledges. So what we can do is to look back how humans do this. They transfer effectively any knowledge they can to the new events or new things. So for example, you even a young kid can learn pretty fast by looking at a few samples. So with that, transfer learning has been very effectively used in the biology domain. And the latest in machine learning field, it's called meta learning, which uses the knowledge learned and to relevant tasks. So with that, I hope to mention the two famous approaches in this for the transfer learning is basically original transfer learning, which you train. There are different kinds of knowledge to be transferred. The most popular one is to train the neural network model with some related tasks and transfer the parameters to target tasks. So that's what the original transfer learning is. And now the latest meta-learning is proved to outperform the regular transfer learning. Basically here, you are training the model with many small tasks, and these tasks including the support set and query set, so you can quickly from the support to the query with many different tasks. And supposedly, once you train the model can quickly adapt to new tasks, then you can use this parameter you trained and with a small sample as a support to adapt the model and get the new prediction model. And so that's what people have been proposing and applying to the bioinformatics domain. And with that, I bring back this TCGA dataset, and you can see they are small samples, but they have a huge amount of features. And then we actually, I want to show you some of the results we have done to show how meta-learning can be effective in applying to this kind of data. And basically, you have all different samples with different cancers, and with small tasks with each cancer, a few samples and prediction, and then you train the model and adapt it to the new model. And I will skip that. So basically, I want to show you the result on this. So basically, we train the model with meta-learning from 31 cancers because there are 2 cancers are too small and the data are not adequate. So and then we validated and we tested in the way. So here are some of the results we did in the TCGA datasets with proteomics data features as features alone, and using more than 7,000 patients over 33 cancers. And we compared with the traditional KNN, the random forest method, and some of the meta-learning method. And this particular meta-learning method called MEMO came to be the best when applying to 3-way means 3 classes you can detect and 5 classes you can detect. So in that you can see the meta-learning can transfer better in detections. And so with different features, you know, the TCGA has RNA feature also, that the result came similar. And so using all the features, actually the result is similar, but slightly lower because of the noises. So that gives us encouragement to apply meta-learning to small sample environment for predictions and transfer learning. So we actually compared substantially between the transfer learning and the meta-learning and see how they behave, how they do better. So I want to show you the result when we compare. So we trained the model using 12 different cancers, and then transfer learning. This is the transfer learning first, and this time we did not use the prediction. We used the survival and death event as a prediction, and then we use 4 new type of cancer, and then see how transfer learning and the new meta-learning memo perform between them. So you can see mostly the meta-learning will transform the knowledge much better than transfer learning. But there are some cases transfer learning does better, and with the data probably more consistent between the new cancer and the 12 cancer we compared. So there are some, but you know, the performance is not far away between those. So these are the performance between transfer. So I think we can claim the meta-learning can do better than transfer learning in transfer knowledge between the different cancers. Of course, this is only transferring between cancers. If you want to transfer between cancer data and some other diseases like Alzheimer, that's a different story. So distribution of the data may be completely different than the transfer of knowledge. Even using meta-learning may not work well. I should point that out. So we did on different data sets also. So I will stop that. But so our main this grant is knowledge guided machine learning. So we want to, so with even meta learning can do better than transfer learning, we still want to be able to integrate knowledge into the model. So what we can do, we can transfer and integrate knowledges. So that's what we are looking at this different kind of ontologies. So the domain prior knowledge in biology is very rich. So for example, you have the disease ontology in the, so for the mimic data sets, you have all this classification of diseases into different hierarchy. And so this hierarchy, we can specify it as a graph and then enforce this relationship into the learning process. Since, for example, the Alzheimer and Parkinson's are closer. And so those, if we specify this kind of relationship, then your machine learning model can do better. So with that, we actually experimented with the mimic data sets, which is a recording of patients in ICU situation. And they have a lot of ICD-9 codes. So we represented this kind of relationship. They have this coding system into the diseases and in hierarchy based on the disease relationship. So we have a graph to design this relationship, and then we incorporate. We have this hierarchy graph to denote the relationship between different classes. And so with that, I will not go to detail of that, but I will just mention this method we published 2020 KDD paper is we designed this hierarchical graph in the disease hierarchy, and then incorporate that into when we train the embeddings, we incorporate that into the model. And so when we train the tasks, we can have the relationship, we can use that as a regularizer to enforce the relationship between tasks. So, and then we can perform better in prediction. So here is the results. When we look at the mimic, they have all different sample, a huge number of classes and examples, but each individual, you have all these individual classes, how they allocate each. And then we compare the performance between all different meta-learning methods, and with our knowledge integrated that graph. And you can see here, when we integrate knowledge into the machine learning model, we do achieve better performance than all other meta-learning approaches. So that shows encouragement when using knowledge into the machine learning model. It does improve the performance. So I just hope to briefly introduce this and as an introduction, and experiments to show the knowledge-guided approach does improve performance. Of course, here in both, let me see what time. So in both scenarios, I use the cancer as a separate training and the mimic as a different training. So they are in different distributions. So, but a question, interesting question is, if I train on the cancer, can I, TCGA, can I use for mimic? I think that performance will not be very effective. I think that comes to heterogeneous distribution of data, how we can train models to deal with the heterogeneous situation. That's a more advanced research topic. So I just have a few minutes before the keynote speaker. So I just want to mention as a conclusion, I think there's still a long way to go, and there are many interesting problems in bioinformatics and health informatics. And I do think the close collaboration between the computer scientist and the domain expert is very crucial and important for effectively applying machine learning approaches in biology. So I will end my introduction there. And so maybe answer, a couple of questions before we introduce the session keynote speaker. Okay, so right here in the chat, there is a question: how does one account for variability coming from different data sources, technical noise, batch effects? So variability come from different data sources. That's a good question here. So you can see if it's the same distribution. So that question I can use this is if it's the same distribution. You can see here we have, when we test the meta-learning memo-based approach on TCGA, we experimented with the proteomic features. There are all, a lot of different features. So proteomics feature alone, this is the result. And RNA expression data, we got this result. Actually, RNA performed slightly better than proteomics here. And then we combined all clinical proteomic RNA, all the data here. This is the result we got. And you can see actually the performance is slightly lower. It's not obvious, but you can see from the ratio. So to answer that question, we look at the data, there are noises and some noises in the whole data sets. That's why the performance is a little bit lower. But you can see in the performance, it is actually pretty effective in incorporating all the noises and in the data set. When we combine all the data. Hope that shows that on here. Oh, we should move on time. So I think we, at the end of the talk, if we have time, we can you know, go back to the questions here. So I see if there's any obvious questions I can quickly answer. I don't been any effect to pan cancer to use. Have there been any effects to do pan cancer to use all in the cancer for cancer that are independent of cancer type? So if I quickly answer that correct, so yes, so if we train the model using TCGA different cancers, so the purpose of using meta-learning is you train with the existing data, you can quickly adapt to new data, especially rare diseases. The rare diseases, you have a few samples, then you can quickly adapt. I think if it's a rare disease in cancer, I think it should be effective if my understanding of the question correctly. What is getting classified? So the 3-way, 5-way means I give you 3 classes, and you should be able to predict it's one of the classes. 5 classes is one of the classes. So that's a typical few shots learning environment. In respect to interpretability, so with graph regularization, that's a good question. You know, the interpretability, it was not a concern in that paper. I think we can do post hoc. If you are familiar with interpretability, post hoc interpretation we can do using this TAdaNet network, but not a pre-hoc, you know, interpretation in the model, because the integration and the deep learning is not it cannot be interpreted in the process, but we can do post hoc interpretation. Okay, so I think I quickly responded some questions. After we have the 2 speakers before the break, I will come back to the questions.